Okay, good morning, everyone. Welcome to today's lecture. It's a great pleasure we invited Dr. George Northoff today. So Dr. Northoff is a philosopher, neuroscientist, and psychiatrist. So his study covers 3 different disciplines. He works in Ottawa, Canada, and he holds a Canada Research Chair for Mind, Brain Imaging, and Neuroethics. He's a fellow of the Royal Society of Canada. His research focuses on the relationship between brain and mind. And he's curious about the question, why and how can our brain construct mental features like self-consciousness? So he developed novel and unique spatial-temporal neuroscience and psychopathology, where time-space are shared by brain and mind. He's also the leading figures in the linking philosophy, psychiatry, and neuroscience. And he developed non-reductive neuron philosophy and the word-brain relation as answer to the brain-body problem. So I guess today he's going to talk about different timescales of the brain and what's their function. It's also a new field for me. So please join me to welcome Dr. Northoff.
Thank you, Cynthia. Thank you so much. Thanks for the nice introduction. So I will. Okay, where did you push the button? Here. Yeah. Okay. So my talk is about timescales, and sorry. Okay. So and it's really I lead you to some hidden dimension. Okay. Okay. Yeah. So first question, of course, what are timescales? Timescales are duration of a process. Yeah, like here I showed you the geological, archaeological timescales are extremely long. But for instance, your heartbeat also has a timescale of 1 second. Your breathing has a timescale of usually more 6 to 10 seconds, and so forth. That's basically roughly preliminary duration of the process. I will detail more. So now I will show you that usually what we do. I assume that many of you are more basic researchers. You try to explain mental features or cognitive features like attention, consciousness, thoughts, and then you assume some computational mechanism here in your models, and that's also what we do in brain imaging and computational, like predictive coding, is probably located somewhere here. But I try to lead you to a deeper layer of the brain, which is usually neglected. And literally, you average out the timescales. When you analyze, let's say, in functional brain imaging, your data, you often don't consider the timing parameters of your experimental paradigm. And that goes in fMRI and EEG. And many computational models also do not really explicitly include timescales. So it's sort of a hidden, I would say, forgotten dimension. However, the background, as you know, particularly in the Chinese culture, of your paintings is extremely important. Because if you have different backgrounds, you see the same foreground in a completely different way. Chinese paintings are beautiful examples for that, much better than Western paintings. So, and my question shows on this how, because I heard that you're mainly a more computational group. I show you a lot of our basic research on timescales, also a lot of computational modeling, and at the end, in the last part, I will show you some clinical applications. So first, what are intrinsic neural timescales? It's a relatively new field. Then what do? They're key for temporal integration segregation of inputs. That what could be potential physiological mechanism, it's recurrent connections intra-region. So when I speak about recurrent connection, I do not mean from region to region, but within the region itself. And then I will show you some example intrinsic neural timescales in the mind, the sense of self, whatever that is. And then I show you 2 very short examples about mental disorders, depression, schizophrenia. So, so first part, what are timescales? I think I already mentioned that. timescales are involved in processing input and task. For a given process, the duration timescale originally comes from physics, where you have a certain half-life of a chemical substance, and same here. And then there's also another definition, I will come back to that later. Timescale is a time window in which prior information can, from ongoing activity or stimulus related, can affect the processing of the new and ionic information. So I will come back now. I focus more on this one. And what is important, that these timescales are already present in your spontaneous activity. Yeah, your resting state, like measured in resting state fMRI, EEG. And how do you measure the damn thing? You put typical, this is one model-free estimation here. You put an autocorrelation window, it's an autocorrelation function. Some of you might know that. We have a signal processing background from engineering, so you put an autocorrelation function. These are the lags, the time lags, T1, T2, T3. And then you basically calculate the correlation of the signal with itself over time across the time. So you correlate your signal at T1 with the amplitude at T2, T3, T4, T5, then the same for T2 with T3, T4, 5, and then you get these lags. And then, of course, the more time lags, the more time passes, the lower your autocorrelation of the system with itself, indicated here. One usual measurement, and often so now this one works here. I think this is the, yeah, here it is. This is a correlation. Usually do it at 0.5, then you determine basically the time window. We invented another one, ACW, and here, this is a package in Julia, which will come out soon in the internal review process. You have different methods of calculating timescales. You have model-free methods, what I indicated here, and you have also a model-based method. There's a Bayesian, very nice Bayesian computation, Zerati et al. 2022 just came out. So you can have different methods and, of course, different ways of calculating the timescale at different purposes. So this will be soon open available for usage also on our website that we are lower run. So now, what about the intrinsic neural timescale? So intrinsic means they are a defining feature of the brain. That's important. So if the time loses the timescales, the brain basically is no longer a brain. And I show later some data, or maybe I don't think I show them here, but you can see them on our website. Particularly when you lose consciousness, you lose basically your timescales. So, and what is important, they also shaped the topography of the brain, meaning the spatial distribution of the timescale. So this is an MEG study, subsequently confirmed by others in fMRI. So there's really, here you can see this, so we distinguish between unimodal regions as the periphery. Here's the blue guys, motor cortex, visual cortex, auditory cortex, and transmodal regions, higher order regions, the red guys, the default mode network, prefrontal cortex, and so forth. And these are three different anatomical distinctions, so just maybe look on this, and you see here I see plotted the two versions of the autocorrelation window down here, after 50%, after 0% correlation, the duration of the process. And you can really see that the unimodal regions, the blue guys, have shorter time windows, shorter ACW than the red ones. Yeah, so that really suggests there's a unimodal transmodal topography that adds to the various findings, really that there's a difference, there's a gradient between unimodal transmodal regions. Here we show you the gradient. You see this quite nicely. This is here specific regions 1, 2, 3, and we really show it's a continuous gradation. It's not just either or. It's a gradient of the timescales becoming slightly, gradually longer from unimodal to transmodal regions. So that really seems to be an intrinsic feature of the brain. It's hugely important for any kind of mental feature and also mental disorders. So we work a lot with that gradient in mental disorders, even in post-mortem studies. We really use it, yeah, because it provides you a whole brain view with a topographic organization, which I think is key for psychiatric disorders like depression. So, first conclusion, timescales are an intrinsic feature of the brain. I didn't really show you all the experimental empirical computational support for that, but just please bear with me. Timescales are an intrinsic feature of the brain. They define the brain and they shape its topography and its dynamics. So now I will show you a little bit why that is, and that's the next part, why that is functionally relevant. So far you can say, I'm not interested in the time scale, not of go home to Canada. But I show you some of the role of these time scales, and actually for those who do biology, these time scales also have been shown in other species, in monkeys. We actually had a study of that with a monkey group in Beijing. So this is really a key feature, basic feature of the brain. So now you remember the two definitions. Now I focus a little bit more on this definition. Intrinsic time scale is the time window in which prior information from ongoing activity. Oh, sorry, that was no, that was the wrong one. This, no. Okay, this one. So prior information can affect, and this of course now leads us from spontaneous or resting state activity to task evoked activity. So the question is also, do timescales modulate task? Yeah, are they involved? Are they really functionally relevant? And just like, okay, this room has only all the same window size, but imagine this room would have different window sizes. You would see the same houses and the green out there in completely different ways. Zoom in, zoom out. That's the same what the different time scales of the brain are about. So now I show you here on the psychological level and then on the neuronal level. So on the psychological level here, this is a meditation. So we study different forms of meditation, particularly in close collaboration with a key center in India. And so there are different forms of meditation. Meditation is not meditation. So you can have, for instance, what is called here the Vipassana meditation, where you scan your different body parts. So you scan you focus your attention on your right ear, on your left ear, on your stomach, sorry, this is the heart, on your stomach and so forth. Yeah, and that means that you need relatively short attentional windows, each window for one organ. And that means that you need a certain temporal segregation, because that's the nature of your meditation. You focus from here, you focus from here. So there's a time component in there. So you have relatively short temporal windows of attention. And the opposite is what is called shunya meditation, where you have extremely long temporal windows of attention, because you are supposed to not focus on anything. You just are supposed to let your thoughts pass by like the clouds, and don't adhere your attention or thoughts to any particular object. Yeah. So there you have very long windows. So, and this is an EEG study, and we said, okay, maybe the temporal windows of your attention here on this level are maybe related to the temporal windows on the neuronal side. So here, the typical autocorrelation windows, this database you can see, and you can measure that in different ways, as I already said earlier. So the typical out here is, of course, zero time lag. Of course, it's one the correlation, and the longer the lags, the less the correlation. From that duration, it tells you something about the temporal windows. Now, and that's exactly what we found. So this EEG study here in the Shunya guys, here, remember this was a completely open meditation. You had very long temporal windows of attention, like here, and you had a very long autocorrelation window. So this is not schematic, it's real data. And in the Vipassana, remember that was body scanning, you have very short temporal windows, because each window includes one body part, and you don't want to confuse your body parts in the meditation. The Indians can tell you all of that. And you see very short temporal windows. Yeah, autocorrelation. So that was some of the idea, and I will come back to that towards the end of my talk, that maybe there's a correspondence between your temporal windows of your neuronal activity in the brain and the temporal windows on the psychological or mental. Yeah, that's the temporal. You mentioned that the spatial temporal approach to a cognitive mental phenomenon. Yeah, that's what we call spatial temporal. I don't show you any spatial examples this time. I don't show you any topographic stuff, but I could show that, give a whole talk about the spatial dimension too. So that's what we call spatial temporal neuroscience. So next one. Now you say, yes, not of all good and so, but this is just psychological, and you didn't really measure the psychological integration. That's true. I will deliver that later. But you say on the neuronal level, you didn't really show me that this autocorrelation window has something to do with the temporal integration segregation of inputs. And that's absolutely right. So what do we do? Here we use a computational model. So that's where the modeling becomes very handy. So this is a large-scale biophysical realistic model. This is based on Menke. Chaudhuri, this is from the Wang group, I'm sure you know him, and published in Neuro in 2015, so it's monkey-based. It's a real DTI-based matrix in there, and you have a distinction between these different regions here. This is, okay, this goes from V1, V4, and so on, and then it goes all the way to the prefrontal cortex with 24C, which is the anterior cingulate cortex. So, and then we stimulated that model with short inputs to V1. So you can see this here, short inputs, very short, 0.25, yeah, 0.25, 0.25. And you could really see, okay, in V1, you have a very nice temporal representation of the input time duration, yeah? The V1 activity in the model, okay, now, okay, that should not happen. Okay, now I do this one. Maybe it's easier with my finger. So here you really see a nice representation of the input from here to here in V1, and then you see a gradually smoothing. Okay, it's another one. Thank you very much. Okay, you're really quick. And see the slides much better. Now you have some enlightenment. Thank you. Yeah, and so you see it's a very nice temporal representation V1, and of course also V1 specialists like an MT specialist like Dr. Song knows this very well. Yeah, and then you see a gradually temporal smoothing of the yeah. So here in 24C, you cannot distinguish between the inputs. So meaning the information related to the input, so each specific input is lost, but it gradually accumulates, and then you see a long temporal smooth input. Yeah. So basically, you can even say it's even going beyond. So the input, all three inputs are still present here in the neuronal activity, though there's no input anymore. That's what we call in our temporal spatial theory of consciousness, temporal spatial expansion. Yeah. Your input is your neuronal duration expands the physical duration of the input. And that's very important for mental features for consciousness. Yeah. So now then we say, okay, here you can see basically the same schematically again. And then we say, okay, maybe it's just the nature of the input. Okay, so we present a long input, same thing again. Yeah, this is a 1.2 second input. You see, it's eternity for the brain. And again, this 24C smoothest. Yeah, and then we calculated obviously also the autocorrelation window of these two regions. Obviously, AC 24C is much longer than the short. Yeah, so that really suggests that this ACW has something to do with temporal input integration. So then here on the right, here again, you see the same for a discontinuous input. Maybe you say it's just the entrainment, which does this. No, it's not discontinuous input. You again see the temporal smoothing. So, and then one of the authors here in this model, who did this modeling, very clever guy. So he came up in the Chaudhuri model. There's a hierarchy index in the formalism. So each region gets a little hierarchy index where it stands in the hierarchy. It's a hierarchy H. You usually overlook this when you look when you work with that. So he said, okay, I set the hierarchy index to 0, and then see whether my hierarchy, my lost topographic hierarchy, which gets an index for each region, then also changes the input process. And that's exactly what it does. Now you suddenly don't see any temporal smoothing anymore. So you know, so temporal integration segregation. Yeah. So that's really here you see it's a very nice example how hierarchy, topographic hierarchy, timescales go along. So now I need to have the other one here for the next one. And so then, of course, what do they do, these timescales? Why are they so important? And you will see they are basically your interface with the environment. So we have a lot of now data will come out and paper will come out from our group where we look exactly for this matching. Yeah, we have a whole line of the timescales of your power spectrum aligned to the timescales of your environmental. Yeah, I don't show this here. You have even we have one paper published a couple of years ago, 50 to 2 seconds intertrial interval, 2 second stimuli, and your brain picks up exactly the frequency range of that very sparse, temporally sparse paradigm. So the okay, this was the wrong one. Next one. So here, so that's a schematic, but again, this is actually supported by a paper which is in revision down here, where we again combined imaging and this is now MEG and modeling, where we combined autocorrelation window plus evoked potentials. So and what you can see basically the take-home lesson of that. So let's say this is the physical duration of your input. Yeah, just focus on this one. And this is here your length of your ACW of your autocorrelation window. So if you have intermediate autocorrelation windows, sort of you may separate this input from these two inputs, but you may not separate these two inputs in your actually evoked activity. Meaning the difference between these two inputs will not appear in your evoked activity, meaning it's lost. Yeah, that's no longer in your consciousness, in your subsequent mental features, but the distinction between this one is still there. Now imagine if you have a very long autocorrelation. Yeah, then you put all three inputs together, you blur them, and you have one evoked activity. And that's exactly what happens when you start losing consciousness. I don't show this here. Your autocorrelation window becomes abnormally long. You blur all the inputs, you put them together, you just have temporal integration, and you don't perceive anything. So you can see this in our paper.
Then how do we select the window length in the autocorrelation properly?
What do you mean by length? So this is the way what I showed you. You don't need to. So if you do it in a dynamic way, of course, then it's a window sliding window thing. But for the autocorrelation window itself, you just calculate it across all data points. Yeah, of course, you need a sufficient number of data points. Yeah, if you do it in a static way. Yeah, so you have your 10 minutes resting state, you have EEG, and you have a sampling rate, let's say, of 512. So then you just do over all those data points, you calculate your autocorrelation window with the length. Now, when you do it in a dynamic way, and I think I'll show you some data later, where we did this in a dynamic way, then of course you need to define what is the window length, and that depends upon your number of sampling points. So if you show me some paradigm and you want to analyze the ACW, one of the first questions I will ask, what is your sampling? Yeah, and with fMRIs, this is of course always problematic, EEG and MEG, and of course shock kinetics. Yeah, so it's always a number of sampling points. Yeah. I think this, my capacity is two guys. Okay, so I hope I can make your case, and I will show you later more data in the mental part about the importance of temporal integrations, in particular for the mental side of things. Yeah, you see how I do this. Usually, you see, okay, you just see the cognitive people just doing mapping their cognitive functions, but I really try to go first into the mechanism of the measures. That's very important because you need to know what these measurements mean. Yeah, you cannot just blindly apply them. Absolutely key. So now I show you a little bit about the recurrent connections. Basically, what are the potential physiological features? And that's why I'm very interested. So here we do computational modeling, but that's why I'm also very interested in cross-species studies, where you can get a much better grip. So I show you one study here, which we did. So this is first data in mice, calcium imaging, open access. So, and what we observe here, so each dot here is one region. Yeah, and these are 5 mice. These are each single mice. And you can see that in the periods where the mice is not running, sustained rest, they call it, it's basically a shorter autocorrelation window than during locomotion. So you see this interestingly for each of the 5 mice, sorry, it's mice. So indeed, that seems to be a robust finding. And also in humans, there are various data, ACW rest versus working memory or attention. You usually see a prolongation of the ACW. However, that might also depend upon the task, whether the task requires temporal segregation or temporal integration. You remember the meditation stuff. Yeah, so it's not always by default. There's actually one study where we show in sleep a shortening of the ACW during a movie presentation in sleep. Yeah, but I don't show this data here. So now, and the next step, then we said, okay, how is this autocorrelation window in the resting state related to the task state? So what we observe, what we did, now we calculated indeed, as you said, ask a dynamic ACW. So we took here windows of 10 seconds, non-overlap. Yeah, and we had a very nice high sampling rate here. So and then we calculated basically the rest, the variability of the time scale of the autocorrelation within the resting state. Why did we do this? Because we wanted to have a dynamic repertoire. You see, my dynamic repertoire is very limited here because I'm just moving back and forth here between this little stand. But if I were constantly moving back and forth from here, my dynamic repertoire would be much larger. So in the assumption that maybe your dynamic repertoire is important, how you will react to input, the dynamic repertoire of your time scale changes in the resting state. So then we calculated a rest task, less locomotion difference, and then we correlated the two, and we got this nice correlation. So it was done here by Yazir Chatal, one of my graduate students. So he calculated the variability of the ACW or time scale here in the resting state, and then linked that to the change of the time scale from rest to task. And what he got was a negative correlation, meaning the higher the variability of your ACW in the resting state, the lower the rest task change. So your dynamic repertoire has a direct impact, and you see this again for all 5 mice, which is quite nice. So and then in the next step, whether the same holds in humans. And that's exactly what's the case. So here is an EEG during rest self story. And you had also, so you had basically resting state, and then you were listening to an 8-minute story. And the story could either be about your own self or about another person. So, and what we observed again, so he analyzed the data in exactly the same way with the 10-second windows. And you could observe a higher variability again, you see a longer ACW in the 2 task conditions in the stories. This was subject-based, this is channel-based. And then here you can see again the same negative correlation, meaning the higher the variability of my resting state ACW is, the lower the change from rest to task. So this is a very nice confirmation in both animals and humans. And now the question is on what are the mechanisms mediating this negative interaction, this transition of the time scale from rest to task, because that's very important how you react to the input. So, and that's where he conducted computational modeling. So now he took yet another biophysical model, because with this human-based Wilson-Cohen model, again, we put in a DTI matrix, the Glaser 268 Glaser regions from the atlas, from the human side, into the model. And it's a neural mass model with recurrent connection. And what we mean by recurrent connection is important. It's really these correlation within the population of cells themselves. So it's not inter-regional, it's intra-regional. That's very important. And these are mainly excitatory to excitatory recurrent connections. So here you see the DTI, diffusion tensor imaging matrix, real data. Here you see the firing rate. And then the nice thing in the model, he could really first replicate the data. So he set the recurrent connections to 1 standard, and he could again, then he stimulated the model, and you could see a resting state, and the ACW is, these are again one region, H dot is one region, and the stimulation, you see longer ACW, and you see again, this is all simulation-based data, you see again the same negative correlation, meaning the more variability of your time scales in the resting state, the lower the change of the time scale from rest to task. So this is basically really replicates the finding in the model, which is always good news, because as you all know, it's not an easy one. And then we said, okay, maybe we now vary the recurrent connection, as I said, the intra-regional recurrent connection within the model itself, and see how this ACW rest, ACW task relationship changes relative to that. So here now the different values of the recurrent connections, and here he plots rest and stimulation. This is all model data, and you remember the ACW is always longer in the task. And so if you have a very low level of recurrent connection, you have no difference between rest and task ACW. Now, you gradually increase your recurrent connections, and you see a really nice differentiation of rest and task with longer ACW in the task. And then you increase the recurrent connection even further, then you see the opposite. So there seems to be, when we put everything together, such an inverted U shape. So meaning you have a certain optimal range in your recurrent connection for the differentiation of rest and task timescales. And that, of course, important, because I don't want to confuse my external input with my ongoing internal thoughts. That probably will happen when you're not thinking about your next thing. Remember, you're not really listening to me. That probably will happen. So that's very important. So you need an optimal range of recurrent, particular excitatory connections for differentiating rest and task states. And why do I say this is particularly important as a psychiatrist? Because in psychiatry, you often have a confusion between internal and external task state, or too much of a differentiation. You can really not really link the two. So this is not trivial. Yeah, and it's also a nice typical feature of a dynamic system. A certain average value of a dynamic here, the recurrent connection, has the best functionality. Functionality meaning rest task differentiation in this case of the time scale. Average is good, extremes are bad, as we said in a larger 2019 paper. So it is really, and that seems to hold for recurrent connection. So I hope I could make the case, and I will show you more modeling data on the recurrent connection later. So I hope that I convince you, okay, there might be a psychological, neuronal, physiological feature underlying. So now the next one, of course, this is basically the homework I have to do then to get to the mental and understand the neuromental connections better. So now I show you some examples here. There's much more on our website on all of this. Show you now the first one, I show you the self. So what is your self? Your sense of self, you define yourself, you constantly change, your body changes, your mind changes, but still you have the feeling that you're the same person. Good morning. So you have a sense of self. And in the Western world, you may know that the self has received a lot of attention. It's usually considered a higher order function, something which is added on to your otherwise non-self cognitive operation. But for us, the self is just how you process inputs. Because I can process the same input in a way which is very much related to me, yeah, self-specific input, so it has a strong meaning for me, or I process the input in a completely way which has no importance for me. So for us, self is not a higher order function, a basic function. Now I show you one example here together with the timescales. So here, this is a classical paradigm. So where you associate yourself. So here, this is a paradigm, one block. The triangle is associated with you. And in that block, then you need to decide whether you see the triangle with you, okay, it's the right one, or if you see the triangle associated with a stranger, okay, it's wrong. And what is amazing that if you associate the triangle with you rather than the triangle with stranger, then you have a processing advantage, meaning your reaction times are faster, your processing speed is faster, and your accuracy is higher. Then in the next block, you associate yourself with the circle. Yeah, so then you and circle. And again, you see the same processing advantage of the self. Faster reaction time, higher accuracy. So the self is relevant on the psychological level. So then we said, how does this work? Maybe this is a called self-prioritization task by Zui developed. Very nice task. And then we said, okay, maybe there's a temporal dimension. So what did we do? Here we present the triangle, then the association of the triangle with the person, we put a delay in there. So meaning you really need to associate this one with this one over a temporal delay. So in that, of course, where timescales come. Yeah, and we had these 3 temporal delays. And I spare you all the details of the psychological finding, but I just go right away. So you had again this self effect, also better for all those 4, for all those 3 temporal delays. And then we could basically make a slope across these temporal delays, how your self-reference effect was preserved. That's here. And then here we calculated the autocorrelation window in these 2 core cortical midline regions, anterior cingulate, posterior cingulate. And you could see that the slope of the curve, the temporal delays, how well people could cope with the temporal delays, depends on your length of your ACW. Meaning, if your ACW was longer, you have a higher self-reference effect over the temporal delays. So the only way for that to be possible that you integrate these stimuli and preserve. So, and we have, I think, that's this here. This is the task I briefly indicated already. So this is the task where we had the story, auditory 8 minutes here, non-self-narrative 8 minutes here, and 8 minutes resting state. So interesting again, we saw this self effect. So the self is not a trivial matter, there's no doubt. One is really surprised how important it is with these psychological effects. So here we did just, this one. So here we measured, so we had a continuous assessment outside the EEG of the story, and we could see in the self, and we measured the distance to which they moved on the screen, continuous engagement. We could really see much more distance moved by the self, higher variability in the movement, and also much more velocity. So your self is important. If you listen to something, you move faster, you move more. It's indeed amazing. And then we calculated the ACW again. We again saw the increase in the ACW. Remember the earlier findings in the mice from rest to task. So this is a quite consistent finding, but not always the case, as I said. And then we also saw a longer ACW in your in the self condition. So this is quite amazing that your self is really associated with longer time scale, and that probably means there's a temporal continuity to yourself. You have, I think I don't show this here. We have various other. So here I show you another temporal integration experiment. So here we did a continuous task. Basically, people had to continuously follow these phases, 10 to 15 seconds of morphed phases. You start with your own phase, you end up with another phase, and at the in the middle, you need to decide when is your own phase terminating, when does the other person's phase start. So, and of course, if you have longer ACW, you see your own self much longer than if you have a shorter ACW, because this requires explicitly temporal integration, and that's exactly what you could also see here. I show you here the, sorry, it's this one, the correlation of the ACW, longer correlation with the self. So meaning the longer you perceive yourself, the longer your correlation window. So that really suggests again that that on the psychological level, delays, timing parameters requiring temporal integration, segregation of inputs are related on the neuronal level to your autocorrelation window. So now, what are the physiological mechanisms of that? So here I come back. You remember the earlier findings on the recurrent connection. So now we pick that up here and do this here. So I don't go into detail about this. So we develop, based on large-scale meta-analysis study, we develop a topography of self regions. So we have different layers of self. Your interoceptive self, mainly, this is all data driven, mainly silent, subcortical, midline regions, insula, related to your inner body, to your interoceptive cells. Then we also do exoceptive, that's basically exoceptive, proprioceptive, cross-modal integration, and then mental self is all your cognition. So, and you see mainly the default mode regions. So now what Khan Keskin did here in this paper, so he compared all these regions versus the non-self regions, because he wanted to see whether there's a certain topography, self versus non-self regions, and how that is related to physiological measures. So that's what he did here. So first, he calculated all self regions versus all other regions of the brain. Yeah, basically self versus non-self. So and the first thing what he observed is we calculated the myelin, the ACW, and global activity. This is basically global functional connectivity in GS-core, the global signal correlation for the fMRI expert. So this is basically a global, the degree of global synchronization in the brain. And you could see here, for the self, you could see lower myelin in the self regions than in the non-self, meaning non-self had higher. ACW was longer. These are all regions together, not separated. Self versus non-self region, ACW was longer. This is no surprise based on our previous finding. And the opposite for the GS-core, it's more stronger in the non-self than, and that's mainly unimodal regions. So then he correlated those three measures. So he calculated the myelin with the ACW, and no surprise, you get a negative correlation, meaning the higher your myelin, the shorter your ACW. And that goes well along with the observation that the myelin is stronger in unimodal sensory regions and less in the transmodal higher order regions. And then you've got the negative correlation, and you've got a mediation model. Myelin impacts GS-core, meaning the global functional connectivity through the timescale. So here you see just on the neuronal level how important timescales are, because they basically mediate the transition from the structure to the spatial functionality. So then he did the modeling, slightly different approach than in the previous one. So he did a Wilson-Cohen model. Again, it's a biophysically realistic model. It's a neural mass model, as I said before. And he focused particularly on excitatory recurrent connection. You can see that here, and the basis tone of neural excitation. Why? Because we assume, based on our prior studies and other basic studies, that these features are particularly related to the ACW. Then he fitted the model to the data, or the other way around. He got relatively good fits. And then he first grouped his empirical, right? So these are the fMRI results. This is all fMRI from the HCP data set. You can see the, and then he split it. It was a very good idea. He split the regions into 4 regions. So transmodal self regions. Over here. Oh, no power in China. Yeah, I try this one. Sorry, I hope I didn't do anything wrong.
Don't worry.
Okay. Okay, so let me do my good old finger. So you see, and then also transmodal self regions, transmodal non-self regions, unimodal self regions, unimodal non-self regions. Why did he do that? Because he wanted to see, is the self, non-self distinction the same as unimodal transmodal? And then, of course, we would have a problem with self. However, that's not the case. To my happiness, it's not the case. You really see a self-advantage in both transmodal and unimodal regions. Yeah, of course, the global tendency is the same, but you see in all 3 measures, you see really that the self is not the same as transmodal, and non-self is not the same as unimodal. Yeah, so that's very important. So topographically, it means that maybe the distinction of self, non-self is nested within the more broader distinction of unimodal and transmodal. So you have different layers. It's like different Russian dolls or your beautiful Russian crystal balls. Yeah, same thing. So the self, non-self is embedded within the unimodal transmodal hierarchy. So that is the empirical data. So, and now, of course, the difficult part, the computational modeling. So these are simulation model results. So the first thing, he said, okay, I fix my recurrence and fix my excitation and let my model fit the data. And see how I can replicate these topographies. So he calls this topographic modeling. I think very clever idea. You can imagine we discussed a lot. So, and you see when you fix the recurrent excitation, you don't really can replicate it. Meaning these two guys are important for the ACW. Now, then he said, okay, I fix my recurrence, but I let the excitation, the basal tone of excitation, fit the model to the data. You somehow get slightly better, but the best one was when you let them both loose and you get this one. And that's really a very nice replication. Usually it's not easy in the model to get that. Everybody who does this knows this. Yeah, so it's quite amazing. Then, so and that really says that maybe this topographic pattern is related to topographic pattern of recurrence and excitation. So that gives you an idea of what the ACW determined both by the topography as well as the recurrence and the excitation. So here we did the same for the GS core. It's not as perfect. We also did a global coupling parameter, which I don't show here, which actually replicated the data much better. No surprise. So that tells you, and this is a summary slide. So you have different layers. You have your physiological layer of the excitatory recurrent connection, basal tone of excitation. Then you have your neuronal systemic layer. You have the myelin, you have the timescales, you have the global synchronization in a particular relation. And now we assume that the self regions, they show increased excitation. They show increased recurrence. What does this mean for input process? Meaning your input is longer processed in the transmodal regions, is really a recursive processing. So, or in the name of the paper, he speaks of an internal echo. And meaning the longer your stimulus is processed, the higher the likelihood it may be that it's associated with your sense of self. And I think that makes sense. So it's basically a basic temporal mechanism. Of course, we still need to show this on the psychological level, that your degree of duration of the process is related to your degree of self-reference. Yeah, but that means you have a temporal mechanism for a seemingly higher order cognitive process. Yeah, let's put a big smile on my face. So now we're trying to do this. And this is very important now. When you have psychotic patients, you have a too long of ICW, you might have too much recurrent processing, which we can basically already infer from our ACW recurrent data. Then you would assume that you have too long processing of your input, meaning you relate the inputs too much to yourself. And that's, I'm not joking, you're resulting in increased self-hope. That's the case in depression, for instance. Yeah. So this is really, it's not trivial here. Now we try to make this point. Okay, so now I show you towards the end of my talk. So here I show you how timescales can be relinked to mental features. And there will be a larger paper of us coming out where we say brain dynamics mediates cognition or shapes cognition, spatial temporal neuroscience, where we summarize all these kind of features. It's really a new view on cognitive neuroscience and also on mental features, which I don't go so much into here. All our consciousness line you can see on our website or in talks or whatever. So here I show you towards the end of my talk some data on mental disorders, because this is another line of our research. Some of those who worked with me already suffered from my spatial temporal delusions. I try to really apply this. So here I show you some schizophrenia data. This is in the ACW is abnormally long in in schizophrenia. This has a tremor here. This is not good. Okay. So you see an abnormally long ACW. You see this in the resting state, particularly in the resting state. The red ones are the schizophrenic. And here we also show yet another measure, which is quite promising in schizophrenia. It's the phase coherence. So it's your intertrial phase coherence. Let's say it's typical oddball or steady state paradigms. Let's say if you play rhythmic music, the phase cycles of your brain align in their timing to the timing of the music or entrainment. And this phase alignment is quite deficient in schizophrenia. You see this here. It's strong in the healthy, so you follow, your brain follows the rhythm of the music here in schizophrenia. Whoop, that was not a good idea. So I have to stay away. It doesn't follow here at all in the schizophrenia. That's a quite consistent finding replicated. So now in the phase cycles are measured on a very short time scale. Yeah, that's basically an event-related parameter. Whereas your ACW is measured over the whole time scale. And that means, so now we were said maybe how the longer time scale of ACW maybe shapes the shorter time scale of the phase. And that's what the phase coherence, that's what you can see here, follows a black line. The higher your phase coherence, the shorter your ACW. Why? If you have a short temporal window, you're temporally more precise, so your phase cycles can align. So this is a correlation we also replicated in other subjects. But look, so meaning if you have longer ACW, you have less temporal precision in your phase cycles, meaning you have lower phase coherence or alignment entrainment to the external stimulus. So in the red line you see in schizophrenia, that's no longer the case. So this is in rest, this is in task. This is a standard task, this is a standard tone, this is a deviant tone, this is an auditory oddball for the experts. And you can see in the red lines, then the red lines are the schizo, they decouple. So what does this mean? Longer temporal windows, remember the ACW was too long, increased temporal integration, this blurs temporal segregation, temporal precision, meaning you have lower intertrial phase coherence. So that exactly this, and they seem to be dissociated, that the fine-grained timing is disrupted in schizophrenia.
Sorry, for this study, is ACW calculated from a person regular?
EEG, channel, ZZ. Yeah. Sorry, I forgot to say, because we constantly switch back and forth. Yeah. So that's a finding we also replicated in other groups. So now you say, what does this mean with respect to the symptoms or the psychological level? Yeah, this is just the neuronal side. So now you want to see what does this mean. So here we had again, remember this phase morphing paradigm. We did this also in schizophrenia. And in schizophrenia, you may know sort of what is called a basic self-disturbance. So what does this mean? You cannot distinguish between inner and outer content anymore. In your delusions, you have an internal thought content, but you say, wow, you look at me and you want to kill me tonight. Yeah, so I project an internal thought content, and I know it's true. That's the certainty that comes with it. Yeah, I'm joking, of course. So, and that's really important because you have this inner-outer thought confusion, and that's the base or hallucination. Yeah, you have some spontaneous activity in your auditory cortex, but you say, wow, somebody's talking to me. Yeah, so this is not true. So we tested for that, and we had due to the Denmark people, they had a whole psychological and phenomenological assessment with semi-structured interview of this basic self-disturbance. So we did this paradigm, and what we could see, which was quite nice, like in the other data, we could replicate the longer ACW. Here, the red ones are the schizophrenia. This is really tricky here. Okay, stay like this. So here you could see the blue ones are the healthy, and the red ones are the schizophrenia. Now, and then we conducted a moderation model down here. So the measurement of the basic self-disorder to negative psychopathological symptoms, subsymptoms of schizophrenia, general negative symptoms. That relationship was mediated by the prolonged intrinsic timescale. So tentatively, because also this was really a paradigm requiring self-other distinction, yeah, inner-outer distinction. Yeah, we could, I think, really say that your inner-outer distinction may be a key factor here mediating the impact of self-disorder on the psychological symptoms. So then I show you, and we have many more studies on schizophrenia, and we really pursue this timescale, particularly in the phase imprecision model. Last year, a good paper in molecular psychiatry on that, and we really pursue this. Also, we analyze data now, schizophrenia in November together with Dr. Song. So now here I show you some of our depression work. So in depression, we have the assumption, so in schizophrenia, we assume the phase is temporally imprecise. Yeah, so it's temporally no longer, it's a temporal precision disorder. For depression, it's for me a speed disorder. So those guys are simply too slow. So it's not just a mood disorder. Of course, it's a mood disorder, but much more. Why? Because when you see a depressed patient speaks very slow and doesn't move at all, and it continues like that. You see no variation, and everything is too slow. No variability. Yeah? Everything is the same. So imagine if I were speaking 10 minutes like this and you would go home. Yeah? You would say no more of this. Yeah? So then you can imagine how the people feel. Yeah. So here it's a study together with Dr. Song and also with Edeling, where we literally measured the speed in the brain, in visual cortex. We literally measured the speed in the brain, and you can measure that with either the different in the frequency bands here, and we show particularly in the faster frequency bands, this is fMRI. In the faster frequency bands of MRI, you see really power differences, meaning the MDD have less functional connectivity here, particularly in the faster frequency, whereas in the slower, it's more or less similar. So that really, and together with various other lines, together with, and then we calculated the median frequency. We literally calculated the speed of the power spectrum, meaning the relation between slower and faster frequencies through what is called the median frequency as an established measure from signal processing engineering. And you could see that the slower your speed here, the faster your speed, the less symptom severity. In all these regions, also here, MT, you're the expert for MT, V1, and the whole occipital cortex. And interesting here also psychomotor retardation. So meaning the slower your movements, the slower your power spectrum. So there you see, again, this direct relation, temporal relation from the neuron to the mental. So then probably, you want to say a couple of things about your task? You're the expert, or shall I? Okay, so it's so here what you can see is motion suppression tasks. These gratings are moving, they move, and you have to decide the direction in which they move. And here you can see, and you have a clear difference how they move and when they detect it, and you can really recalculate. Dr. Song and Yongao are the experts for that. You can really calculate the distance, motion distance, duration threshold, and you can really see that the depressed patients are really different, meaning they move too slow. The duration is longer. I don't show that here. The duration is longer. Why? Because they're too slow. Yeah, so they need longer to perceive the same thing. So with that, I come to the conclusion. I show you a couple, 3 or 4 slides in the conclusion. I hope with this part, I could somewhat convince you that timescales can connect neural and mental features. As I said, there's many, many more studies on our website, also in this year or last year, that you can really see that we can make a link with that. So why does Northoff emphasize the timescales so much? You can say Northoff, you're schizophrenic, you're delusional. And but timescales are key. Just as a thought experiment from a recent paper of ours. So imagine, so music has a variety of timescales, slow and fast. Now, if your robot has only the faster timescales, the robot will always dance too fast. If your robot has only the longer timescale, it will always dance too slow. So you need a mixture of different timescales to optimally align to the rhythm of the environment. So instead, but true, this is literally the case in manic patients, bipolar. They're too fast. See a manic patient, he runs around like crazy. Yeah. For the manic patients, we are too slow. They don't perceive themselves as too fast. Yeah, because for them it's a default. In depression, it's the opposite, like this. Yeah, literally too slow. Of course, it's not like this, but it's a shift of the time scale. Yeah, and as I said, we have a whole line of really showing the speed disturbance in depression and bipolar disorder. So now on a more biological level, this is a schematic slide, but this is something I would love to do, in cooperation with people who work with other species, that I expect that your environmental contact, your input processing, strongly shaped by the kind of timescales you have. Yeah? If you have a very small timescale reduction, then you will have a very small interface with inputs from the environment. Because it's only those timescales of those inputs for which you have your own timescales you can pick up. Otherwise, you can brain process. A typical example in the consciousness debate is always about what is it like to be a bat. A bat has ultrasonal. We cannot process ultrasonal, meaning the bat has a different interface with the environment than us. Yeah, so that's why I consider timescales as so important, and I would love to run some experiments with these different species, showing that or reanalyzing some existing data with timescales. So I always compare that to the windows. Yeah, so your brain has a variety of different temporal windows. Yeah, and through each window you see something different. If you have a global window, you see the whole scene, but you don't see the detail. If you have a smaller window, you see a lot of details, but here you don't even know what kind of landscape that is. Here you could also be in a living room. You don't know. And your brain has apparently all these different timescales through which it approaches the environment. Yeah. So that's developed us to, based on a variety of others and own data, what we call the common currency, or here very recent this year paper, taught the theory of common currency, so that we say you have similar temporal features in your neuronal activity and in your mental activity. So here, for attention, it has been quite well shown, neural fluctuations are related to attentional fluctuations in corresponding frequency domains. Yeah, here we have another paper in PNS coming out on the time scales of thought. Completely crazy. One of my grad students came up with the idea that time scales longer and shorter can distinguish different kinds of thoughts. It is absolutely completely crazy, but it's really here, psychomotor regulation related to the delays in functional connectivity. So I could show you the same on the topographic side, which I refrained from, but it's not a one-to-one correspondence. It's not that you need to have the same time scale. Yeah, you have a shorter time scale is nested within a longer time scale, meaning a shorter time scale of your mental state can be related to a longer time scale in the brain. And we have another paper which just came out where we really show it's a Granger causality, but time-lagged Granger causality from the speed, neural speed, to the speed of your emotional assessment. So it's what we say a complex correspondence. And that's very important for psychiatry. I already indicated that, so we have the same spatial temporal approach. Apply this to psychiatric disorder. As I said, mood disorders are for me speed disorders, but then, for instance, anxiety is about regularity, and they have higher entropy. And we can show this in the brain, in the breathing, and in the heart rate of anxiety patients. Schizophrenia is for me a temporal imprecision disorder. Remember, these phase cycles are less coherent. So with that, I come to the end. I hope I could make somewhat of a case that timescale may connect.
Sorry, could you please explain more about how to calculate ACW from EG or MEG data?
Take some time. You need a time scale for that. Okay. Faster than I thought. Okay. Yeah. So basically, you take your signal points T1, T2, T3.
So the y-axis is the correlation of
The y-axis is the correlation of the signal with itself.
So the signal with
Itself. It's a temporal cross correlation. So Okay. So these are your different time points.
Yeah.
Okay. So now I correlate. This is T0. This is T6.
So it's the correlation between T0 and T6 of the engineer.
T1, T2, T1, T3, T1, T4, and then the same for T2. That's the curve. These are the legs.
So
Yeah. It's a temporal cross correlation, autocorrelation. When you look into signal processing engineering, that's where it's comes from.
now how do you choose the time lag between T0 and T1?
That's my sampling point. Remember, the number of time points are very important. Yeah, your sampling rate. So the fMRI sampling rate, if you get a sampling rate of 2 or 3, it's catastrophe. You will see this in my face right away. Yeah, because then you have, let's say, in a 5-6 minutes resting state, you have 168 volumes. It's not very time. Yeah, you can do it, but don't make me happy. Yeah, because that's so the disadvantage of this unique time points. But ultimately, come on, if you do simple function connectivity, it's the same things. Yeah, it's also time series based correlation. Yeah, but it's much more simple. So and yeah.
Question. So, suppose the input or the stimuli also has, suppose it's also a time series data, and it also has some sort of autocorrelation.
It's the input.
Yes, the input. So, do you think there's some kind of interactions between the ACF of the input and the neural activity?
Yeah, it's remarkable. I don't show those results here, but you can see this on other talks in the website. That how much the brain follows the stochastic distribution of it. And part of that is, of course, music. Yeah, you have a music, a melody. There's an autocorrelation. And that's exactly what we want to do now. We start now analyzing the input itself. With the same measures like ACW or another one is power exponent state frequency now. Same thing. Yeah. So it's remarkable what you see. Your brain tracks the stochastic structure, the dynamic patterns of the input through its adapting its own dynamic. It's for me, it's mind-boggling. Yeah? So you're not here. So even now, you cannot avoid that your brain follows my movements. No conscious control. Yeah, and I predict if I do this another 2 minutes, your brain will show a peak in its power spectrum in the frequency I'm doing this. So you either better leave the room or you have no choice. Yeah, it's remarkable, and that is relevant for consciousness. So we have papers where we show when you don't have this power spectrum peak aligned to the environment, you lose your consciousness. Yeah, and now we're trying to do the same for second.
Thank you.
So very interesting too. I also noticed that the ACW of resting state and the task are quite similar in terms of topographies in both
topographics.
Yes, I noticed the one figure, right, in your presentations. How do you comment on this? Do we have the dominant intrinsic time scale in both resting state and the task?
Your task is just, as Rector said, at best 5% incremental. Yeah, the problem is to current neuroscience that you average out to 95%, yeah, by doing event-related analysis, and then you overextend the 5%. So of course it's an intrinsic feature, and we're working on that. This is works. If you really look how the topography of your ACW shapes your task states. Yeah, and of course, let's say if your ACW and visual cortex is already too long, of course I showed you the model. You put the inputs together, you cannot separate. Yeah, so absolute key. And that's why in our papers, you will always see, and the ones who work with me, they'll always see rest-task interaction. Yeah, I know the cognitive people say, no, there's no rest, put rest to the rest. Yeah, was a TICS paper in 2019. Yeah, because now the rest is internal cognition. Yeah, and the task state is explained. Come on, the brain is not the same. Yeah, and it's really the resting state is the basis for both internal and external cognition. Yeah? That's why we develop a baseline model of cognition, 2022, do it as a baseline. Yeah, That's why the resting state, and because your resting state has all the intrinsic properties, your topography, your dynamic stuff, and that shapes your task. Your task is just a modulation, max 5% of them. Yeah, energetic modulation goes back to right, and you can see that. That's why I always do this rest task. Ultimately, you want to have a readout from the resting state, its dynamic repertoire, how you will react to the task. That's what my idea is also for the psychiatrist. That's
Thank you.
the resting state. That's the whole approach I'm doing. I developed so that through the spontaneous activity. Yeah, because I was asking myself, what features are there in the spontaneous activity that could shape cognition and serve as a baseline? Because it's like the floor. The floor is the baseline for us in this room. Yeah. If the floor were crooked, everything would be different. That's your brain's resistance.
I also have a question. Very interesting view from the time scale of the brain. Because my background is from neural circuit, I wonder how much do we know what is the neural circuit or structure basis underlying different time scale. For example, sometimes they respond faster with a higher spatial resolution. I wonder what is the circuit basis behind it. Do we know anything about it?
Not really. You see, my desperate attempt to go into the recurrent connections there. That's definitely, it's clear, it's intra-regional measures. This other paper, which is in revision, where we show it's also maybe certain cortical layers. Yeah, it's maybe the upper cortical layers, superficial pyramidal cells, input layers 1 and 2. But again, we don't really know, and definitely not, I cannot go as far as you can.
Question? If no question, we prepare some little gift. One is from the Hangzhou Laboratory, so it's a sign for your lecture with your name. And we also have a gift from.
Now I finally know what my name is. Thank you.
We also have a gift from the Brain Institute. So this is a introduction of our institute, and so you can know more about our institute. And also, this is probably more useful. It's a fast dry T-shirt.
Very nice. Okay.
So you can. Well, this is the name of our university.
Okay.
In the
I will
back.
carry it into the world, and I hope that nobody asks me what it means.
So this is the back is the name of the university, Zhejiang Daxue, Zhejiang University, and this is our institute name, Brain Institute, in Chinese.
Thank you so much. You forced me to make it. Thank you so much.
Right. Let's thank Dr. Notov again.
Thank you. Like how time series firing rates.
我说我正在找，我以为
That would really be fun going to.
Yes. Yes.
所以说怎么
If you want to do this, let me know.